Okay, so reviewing from the last class. Here's the problem for you to think about. We finished with the dihybrid process. So here are 3 questions for you to see if you can answer. You can talk to other people if you want to. Answer is not on your phone, I guarantee it. Okay, first one. If the tall allele H is dominant over the short allele and green seeds are dominant over yellow, what would be the genotype of a dihybrid plant? A dihybrid plant would have what genotype? Okay, dihybrid meaning what? Both traits are hybrids, so they're going to be homozygous or heterozygous. Heterozygous, so what's going to be the genotype? Big H, big G, little g. Okay, what would be the alleles or gametes that plant could pass on to the next generation? This is what we practiced with FOIL, which you'll practice in lab, and we'll keep talking about it until, remember, if FOIL helps you, that's great. If FOIL doesn't help you, don't worry about it. This plant could pass on, if we talk about the firsts, big H, big G would be one gamete. Second gamete, the outer ones, big H, little g. The inner ones would be little h, big G. Or the last ones of the pair would be little h, little g. What would be the phenotype of a plant that is big H, big G, little g? It's going to be tall because it's homozygous dominant for the tall traits. And it's going to be green. So I thought it's weird that yellow was dominant over green in the example we did. And I went back and looked at it, and green is dominant over yellow. So I don't know why the book even has an example where yellow is dominant over green. I know that you weren't up at night thinking about that, but I was like, why? This doesn't make sense to me. If you were, good. You can email me in the middle of the night because I'm thinking about it too. Okay, so we talked about monohybrid crosses, we talked about dihybrid crosses, we talked about test crosses, and we went through some Punnett squares. We talked about the 2 laws of inheritance that Mendel came up with. And now we're going to talk about dominance patterns because, surprise, it's not as simple as what we talked about. Okay, so some traits do follow these complete dominance patterns where what the dominant allele fully covers the recessive allele, and it's nice and pretty to make the Punnett square, and everything works out nicely. But as is life, things don't always work out the way that you would like them, the simple way to work. So we have some other dominance patterns that we have to talk about. So the first one that we're going to talk about is incomplete dominance. So again, thinking about this in comparison to complete dominance, what do you think it means if there's incomplete dominance? So in complete dominance, the dominant allele covers the recessive allele. So in incomplete dominance, it doesn't cover it totally, but it does cover it a little bit. So in this case, neither allele covers the other one fully. So we do get what we would call an intermediate phenotype. Meaning that we get a phenotype that looks like it's somewhere in between the 2 alleles. It's still not blending.
Whoops. It's not that the two alleles are combining with each other and making a whole new allele. It's just that both alleles are expressed partly because they're not covering each other up. So it's not that we're changing the rules of blending or not blending. So the classic example of this, because there are classic genetic examples, the classic genetic example of this are snapdragons. Because in snapdragon flowers, you can have a red parent and you can have a white parent, and then the offspring are pink, which is an intermediate phenotype. When we write an incomplete dominance phenotype, Punnett square, we do it a little bit differently. Because you don't have a true dominant and recessive allele, we don't use capital letters. So it's not like we're like, oh, big R, little r for red and white. We use, and this circle is not really big enough for this. So I'm going to write it here. We use a system that shows we have a red allele and a white allele, and they're not completely dominant. I don't know why we use the letter C. I meant to look that up and I forgot. I don't think so, because I don't know. In blood typing, we use I's. I don't know. I don't know why we use different letters, and I should look that up. So if we take a red parent that is true breeding red and we cross it with a true breeding white, they're still going to have two alleles for the trait. This parent can only pass on the red, this parent can only pass on the white, so that we know each offspring is going to be what? What are they going to get? This parent can only pass on what? Red, and this parent can only pass on. So they have to have one of each. So half of their alleles are going to be CRs, half of them are going to be CWs, and again, you're going to see pink. And the reason that we know that this isn't a blending thing where we've made some kind of new pink gene is because if we take the F1 generation and we let them self-pollinate, or we cross them, we end up in the F2 generation with one pink plant passing on CR or CW, because those are the possible alleles. The other pink plant passes on CR or CW. So we end up with these red plants. We end up with two pink plants, and we end up with one white plant. So the genes have not gone away. They haven't like turned into something new. They have just masked each other, sort of. So what would be the phenotype ratio for the F2 generation? So the genotype ratio we would write is this. What would be the genotype ratio? How many are CR? Okay, let's have more than one person give the answer. Yes, one. You can hold up your fingers. How many are CW? Two. And how many are CW? There's only four, so we can do that. 1 to 2 to 1 would be our genotype ratio. Oops, sorry. For our phenotype ratio, we have to include all three colors, because there are three different phenotypes, even though we're only looking at an allele that has two possibilities. So how many red do we have? How many pink do we have? And how many white do we have? It's a 1 to 2 to 1 ratio, would be the expected. Is this always what happens? No. Okay, we'll see next chapter when we talk about sex determination. You have a 50-50 chance of having a boy or a girl. You all know one family where they have like all girls or all boys. I taught a student who had, their family had 6 girls. And she got pregnant with twins, and it was a boy. You know, you're excited. But one of the twins died, but the other one was a boy. So, but like, you know that it doesn't always happen that way. So if you really grew snapdragons and there are 4 flowers, you're not guaranteed to get 1 red and 2 pink and 1 white. It's not a guarantee. That's just an expectation. This is a prediction we can make, a hypothesis that we can make. So incomplete dominance is a possibility of the different ways that we can inherit traits. When we talk about blood types, we're going to talk about 2 other kinds of inheritance patterns. that are included in blood types. Those of you who had lab yesterday did your blood type. If you're brave enough, those of you who have not had lab yet, guess what? You get to bleed in lab. You can find out your blood type. So this has two different inheritance things that are important. Number one, there's the genetic concept of multiple alleles. So up until this point, everything that we've talked about has had one possibility. There's one or the other. There's only two possibilities for each allele. There are traits that have more than two alleles, and in the case of blood typing, there are three possible alleles. There's I big A, I big B, and that's a little I. And little I. Okay, so there's three alleles. And here about this is you still have some complete dominance in this case. So in this case, we see complete dominance of either of the capital I's over the little I. So there's complete dominance of that. But if you have both capital I's, there's not necessarily complete dominance. There's another trait or another inheritance pattern that we also see in blood typing. And that's the idea of codominance. And in this case, both alleles show and affect the phenotype. So the three alleles for A, B, O blood groups are IA, which codes for an antigen, a carbohydrate that's attached to the red blood cells. That isn't really a triangle, but we always use triangles. And then IB, which codes for a different carbohydrate that attaches to the blood cell surface, which we represent as circles. And then little I, which means no carbohydrates, markers on the surface. And again, we're presenting blood type as a simple, oh, you're AB or O. There's all kinds of other factors that affect blood type. There's other proteins and carbohydrates that affect blood type, but this is the simplest one that we're all more familiar with, I should say. So because we have both multiple alleles and codominance, we have more than two different phenotypes that are possible in the offspring. So you can get different types of blood, which again, you're probably somewhat familiar with. I have A blood. Okay. There's B blood. So A has the A antigens, B has the B antigens. What do you think this is? AB blood. And how about this one? Oh, there's nothing on there. It's all alone. It's just a note. Okay, so you have one of these types of blood. If you have A blood, you have two different possibility of genotypes. You can be big IA, or you can be IA little I, because there's complete dominance there. If you have this blood type, you're still going to end up with the A antigens. Same thing for B. What would be the possibilities for genotype? You could be homozygous dominant, or it's not really homozygous dominant, it's just homozygous, or you could be heterozygous, IBI. How about AB? What are you going to have? You have to have a gene that codes for the A antigen, and you need a gene that codes for the B antigen. So there's only one possible blood type. How about O? Yep, double little I. So homozygous again. And we would call that homozygous recessive because it's recessive. So no antigens. So when we make a Punnett square for this, I'm going to make you make one. I have 4 kids, so I was really excited because I've got like a Punnett square for every genetic trait. I can like figure it all out. But blood typing was not very exciting in our family because I have A blood and my husband has A blood, and guess what? All the kids have A blood. So it's not any fun. So let's pretend we have more fun blood types. And we have a parent that's AB and another parent that's O. So take a minute and see if you can fill in that Punnett square and see what would happen in this case. And go ahead and do like a phenotype ratio. Remembering there are 4 phenotypes, A, B, AB, and O. Which you got on last.
Yes.
Because it shows dominance and more cells in there, so I got the eyes in there too. That makes it a little bit weird. Okay, so let's think about the parent that's AB. What could they pass on? What alleles can they pass on? Remember, we're segregating them so they can pass either an A allele or a B allele. And what about the parent that's O? What can they pass on? Only I's. So as you fill that in, again, you're going to write the more dominant one first. So if we do our phenotype ratio, we'll do A to B to AB to O. How many of the kids would have A blood? What does this kid have? This kid would be A, this kid would be B, this kid would be, and this kid would be. So we have how many A's? How many B's? How many ABs? How many O's? So you have a parent that has AB blood and a parent that has O blood, and the kids look totally different than the parents in terms of blood type. It's not possible for an AB parent with an O parent to have a kid with AB blood, or an O parent to have a kid with O blood. It's my favorite Punnett square because I think it's interesting that the kids can't look like their parents. So other things to think about when we're talking about genetics. We're talking about this all like one gene affecting one trait. One trait affecting one gene affecting one trait. When we talk about these traits, and you'll see this in lab also, a dominant trait in life does not always mean it's the most prevalent. Okay, just because a trait is dominant doesn't mean it's what you see the most in a population. Your book gives the example, and you'll see this in lab too. Not sure when I put those in haven't had lab, but having 6 fingers or 6 toes is actually a dominant trait. Polydactyly is dominant even in humans. Okay, I don't know if any of you have 6 fingers or 6 toes. If you did, you probably had an incomplete digit that is removed. It's usually removed pretty young because it's not functional. It's not like an extra functional finger. It's not like a Princess Bride kind of thing where you've got the 6 fingers and you know you're looking for the 6-fingered man. It's not that kind of finger. It's like a little nub often that's removed, but that's dominant. And you may know somebody who has been in that situation, but the majority of people that you know probably did not have 6 fingers and 6 toes. So even though it's a dominant trait, that allele is not most prevalent in the population. And you may see that for some other traits in lab too. Usually it's a little closer in that like some things might be like 60-40,
It's not as dramatic as polydactyly, but just because it's dominant doesn't mean it's the most
prevalent in a population. A second idea that we have to talk about, we need to talk about, not have to in a bad way, is pleiotropy. So this is the idea that when we talk about a trait, one gene can have multiple effects on the phenotype. So even though it's one gene controlling one trait, that can end up having the effects on lots of different traits in the person's body. So the phenotype is affected in more than one way by this one gene. So some examples of this, we usually talk about our diseases, genetic disorders that cause significant effects in the body. One of which is CF. Anyone know what CF stands for? Cystic fibrosis. So what happens to people who have cystic fibrosis? So the most common thing that we think about is the lungs filling with fluid and having to be able to get the fluid out of the lungs. There's other effects too that we don't talk about as much. Cystic fibrosis is relatively uncommon. It's affects about 1 in 2,500 people of European descent, which to me still sounds like a lot of people, but it's not a significant number of people. In terms of, you probably don't know 1,000 people that have cystic fibrosis. If you know someone who has cystic fibrosis, it's very significant in their life. This is, in this case, the normal allele is the dominant allele. So this is a recessive allele. So the normal allele F codes for a membrane protein. And that membrane protein allows the transport of chlorine ions. So it's allowing the transport of chlorine ions between the fluid, the intracellular fluid, and the cell itself. So the movement of chlorine ions is controlled by this membrane protein. So the normal allele allows for normal movement of chlorine protein. If the person has the recessive allele, little f, they have missing or defective membrane proteins. And again, we're talking only about one specific membrane protein that is allowing the transport of chloride ions. Because the chloride ions aren't moving properly, they end up with more fluid. Which will attract, which turns into mucus basically. And the mucus affects not only the lungs, but again, that's the most common one that we talk about. It also affects the digestive tract, the pancreas specifically. So it affects breathing, it affects absorption of nutrients. So again, this one gene controls multiple phenotypic effects because the mucus can build up in multiple places in the body. And it's a genetic disorder, so we don't have cures for this yet. Okay, we're going to get into gene therapy in a couple chapters. That's a possibility, but it's not a reality yet. Another example is sickle cell disease or sickle cell trait, and we'll talk about that. This is more common in African population, so it affects about 1 in 400 African Americans, which is a significantly larger number than cystic fibrosis. And again, it's a pleiotropic effect where this one gene has multiple effects on the body. So in this case, we're talking about a gene that's controlling one amino acid in the protein hemoglobin. So hemoglobin is the protein in your blood that's carrying oxygen. There's a mutation in sickle cell that affects one amino acid. So we're talking about hundreds of amino acids long. One amino acid gets changed. Because of that change in that one amino acid, there's significant effects. So some people who are homozygous recessive have sickle cell disorder or disease. I think we say disease. I should know. Individuals who are heterozygous have what we call sickle cell trait. We also sometimes use the term carriers. So they're carriers of the trait. And individuals that are homozygous dominant are not affected by it in any way. So people who are homozygous recessive have all sickle cell hemoglobin. HB is the abbreviation for hemoglobin. So they have all sickle cell hemoglobin. What that means is when the body has low oxygen, the protein starts to aggregate and it makes the red blood cells go from being a nice donut shape to being shaped like a sickle or crescent moon. So this happens more regularly. It can be triggered by low oxygen. Sometimes it's triggered by low temperatures. So when the red blood cells start to sickle, then they don't fit through blood vessels as well. So you have places in your body where the capillaries are small enough that we're talking about one cell can go through at a time. And places where blood vessels branch, your normal donut shaped ones can kind of fold up and move their way through, but the sickle cell ones just kind of stick together. And so they start to block off blood vessels. So when we're blocking off blood vessels, that means that they're tissues that are not getting oxygen. So there's a lot of phenotypic effects of this. So when we have low oxygen and we're in a sickle cell crisis, it causes pain because your blood vessels are physically blocked off. It can cause exhaustion, fatigue, because you're not getting oxygen. And if you can't do oxygen, you can't do what? Cellular respiration. So no oxygen, no cellular respiration. So fatigue, weakness, and it can even, depending on how serious the crisis is, it can cause a stroke because you could be blocking off blood going to the brain. So it can cause a stroke or paralysis. So one amino acid in one protein is changed, and you're talking about a person who has significant phenotypic effects in their whole body for their whole life. Because again, it's a genetic disorder. There are children who get blood transfusions regularly to help eliminate sickle cell by eliminating sickle cell red blood cells. And there are drugs that can help treat it, but again, no cure for this yet either. The other interesting thing about this is individuals who are heterozygous are actually have a resistance to malaria. So malaria is a parasite, a blood parasite, and so it doesn't populate sickle cells. So people who have this heterozygous trait, they have half of hemoglobin that is normal because it is not a complete dominance, and they have half of sickle hemoglobin. So they have some hemoglobin and that's normal and some that is sickle cell. And so they end up with a reduced frequency of malaria. And if you live in an area where there's high malaria, this is a benefit, which means you're more likely to survive and be able to reproduce and have more children and pass on that sickle cell gene to children. So there's a benefit to being heterozygous, but again, more likely to pass it on to the next generation, which is one reason that the allele has a higher prevalence. People who have full sickle cell disease have a reduced life expectancy, not that they can't have children, but they have a reduced life expectancy because of sickle cell crises. And so there's a significant disadvantage to being homozygous recessive, but there is some advantage if you live in a climate with malaria of being a carrier of the malaria of the sickle cell gene, which I think is interesting. My uncle is a hematologist and he works a lot with sickle cell patients. I almost made you watch a 14-minute video of him talking about sickle cell, but I decided we would not watch a 14-minute video talking about sickle cell. So both of those are examples of pleiotropy where one gene is having multiple effects on the phenotype. Some other things to think about. We talked about with Mendel, the idea that genes will assort independently of each other. And so that kind of gives the idea that like it doesn't really matter one gene doesn't affect another, but sometimes one gene does affect another. And that's what we call epistasis. So your book gives a classic example because who doesn't like labs? Maybe you don't like labs. They shed a lot, but they're cute. When you talk about lab code color, there are people who have black labs, there are people that have chocolate labs, and there are people who have golden labs. And this is really the effect of two different genes. So we're talking about one gene that's affecting the expression of another gene. So there are two different loci on the chromosome, but one gene affects the expression of the other gene. Epi meaning like above, so having this idea of having control over the expression of another gene. So in this case, we have black labs and chocolate labs and golden labs. And so we have two genes here. One is the gene for coat color. And there you can see there are two different forms of it. So it's a normal gene in that there's two alleles. There's the big B form and the little b form. So big B codes for a black coat and little b codes for a brown coat. So that makes sense if you're looking at this. We've got black, we've got homozygous for the coat color, heterozygous for the coat color, and they're all black. That makes sense. And little b is recessive, and so this one's homozygous recessive, and it's brown, and that makes sense. But then we get to these golden labs down here, and something weird is going on. Because this one has two big B's, but it's golden. So it doesn't have that black phenotype. It's golden because we've got the other gene. I should put the B in. The E and the little e gene. And these two together affect how the color is deposited. So how those pigments are actually deposited on the hair of the dog. So if you have the big E gene, like this dog does, you've got the gene for the black pigments and you've got the gene for color deposition. So you turn out to be a black colored dog. In this case, this brown dog has the little b genes for a brown coat and the big E gene saying color should be deposited on the hair. So you get a brown dog. But here we've got the two big B genes, which would make you think the dog should be what? Black. But we've got two recessive genes here, which says, hey, the color isn't going to be deposited on the hair. So I guess I should. So if you have the dominant gene, it says that the color will be deposited on the hair. But if you have the recessive genes, it says the color is not deposited on the hair. So it doesn't matter that the dog's body is making black pigment or brown pigment. There's no way for it to be deposited on the hair. So this E gene has epistatic control over the B gene. So the genotype of the E gene controls the phenotype of the B gene. Does that make sense? So this dog is recessive, homozygous recessive for the color deposition. It's always going to be a golden one, whether it has the genes for brown or the genes for black, it doesn't really matter because this E gene for color deposition has epistatic control over the coat color. Anyone here have a black lab? Chocolate lab? Yellow lab? We only have one. We have what? So you have a dog that has little e, because there's no color being deposited, and you have a dog that has little b, and at least one big E to get brown. Okay, last thing we're going to talk about. Oh, no, two more, sorry. Some genes are also, or some traits are polygenic. So this is like the opposite of pleiotropy. This is where more than one gene affects, I need to just. More than one gene affects the phenotype. So it's not just that there's one gene that controls eye color. So in humans, we often talk about eye color and how eye color is passed on and, oh, you have brown eyes like your parents, or you have blue eyes and your parents have brown eyes. And we act like there's one gene, but it's a multiple gene kind of thing. Height is another thing. There are multiple genes that control height. So you can have two short parents that produce a tall kid somehow, or two tall parents that produce a short kid. Okay, both my parents are 5'8" and 5'9", and my mom said she prayed that I would be 5 feet tall. And I'm 5'3.5". I said, you should have had more faith and you should have prayed for me to be 5'6", because I would really like to be 5'6". But both my parents are tall, but there's multiple genes that affect height, and both my grandmothers were short. So I ended up with the short genes from both sides that made me shorter. I'm average, almost average for a woman, but still kind of short. So again, just having the idea that there's a continuum, and this leads to a continuum. I think I spelled that right. Of phenotypes. Okay, it's not that I have a parent that's 5'8" and a parent that's 5'8", and all the offspring are 5'8". There's a continuum. My sister's 5'6". I'm 5'4". My 4 children are going to be extremely different heights. I have 1 normal child and 3 freakishly small children, and I don't know where they came from, but they are. You know, when you go to the doctor and they look at the growth curve, like my first child, they're like, oh, she's right on the 50th percentile, maybe 75th. And then my son came along and they were like, well, he's kind of on his own curve down here in the 3rd percentile. And they don't look like siblings that are 15 months apart. They look like siblings that are 3 years apart. And then my son hates it, but eventually he might catch up. I don't know, maybe not in weight, maybe in height. And then there's also the idea, and we're not going to get into the psychology of this too much, but this idea of, are you who you are because of your genes, or are you who you are because of your environment? And I think probably would agree there's probably some of both. And genetically, even we would say that too. Oops, see, I don't need that yet. Your environment affects your phenotype too. So if you're like me and you're pale and you go outside and get sunburned, okay, the environment is going to affect your phenotype. Some people who have darker skin are not as affected by the sun, but environment still affects phenotype in lots of different ways. The example that's here, it's covering up my words here, is hydrangea plants. Hydrangea plants grow different colored flowers based on the pH of the soil. So if the soil is acidic, they will make blue flowers. If the soil is basic, they will make pink flowers. So it's not that they have different genes to make them blue or pink, it's the pH of the soil and the way they uptake aluminum that will change their pH. I mean, sorry, the pH changes their phenotype. Okay, so this is something else that you're going to do in lab, and we're going to do a little bit here too. We can take and trace a trait throughout a family over a period of time, looking at a family tree, and we call that a pedigree. So looking at a specific trait over a family for multiple generations. This works when we're using a trait that has simple Mendelian inheritance. So meaning it is passed on this in a simple Mendelian way. We're not talking much about incomplete dominance and multiple alleles and all that kind of stuff. This is Mendelian human traits. So we're looking for, this is going to show patterns of inheritance in a family. And it can be used to make predictions. Like, are you going to pass this on to your offspring? So you're practicing this in lab as well, and we're going to do a couple together. And so the first thing you need to do when you look at a pedigree, so we've got a family chart over here. Squares are males and circles are females. Affected males are filled in and affected females are filled in. If there's a line between them, they're producing offspring together, and these are showing you the offspring in birth order, always left to right. So the first thing you have to do is you have to determine the pattern of inheritance. So when you look at a pedigree, you need to figure out, is the trait that you're looking at recessive or is the trait that you're looking at dominant? Because that's going to help you figure out what is going on. So how would you determine if something was recessive or dominant? What would be a clue that maybe something was dominant? So if one or both parents have the trait and the offspring have the trait, that's usually an indication that it's dominant. Yes. Okay, if you want to, usually I start to look at patterns to see first if it's recessive, because I think that's an easier way to look at it. But dominance, usually what you see is it's present in every generation. Recessive, you might see that it skips a generation, meaning you have two parents that are unaffected, but their offspring is affected. So if unaffected parents have affected offspring. And when we say affected, we could be tracing something like, this one is showing widow's peak, which isn't like a condition that you're affected with necessarily. Affected just means you have it or don't have it. So if it's a recessive trait, you're looking for unaffected parents that have affected offspring. If it's dominant, it's usually present in every generation. But this example over here is going to make it that not look like it's true. But once you've figured out what you think the pattern of inheritance is, you need to fill in what you know. We're going to start with one here where I'm just going to tell you. So widow's peak, you're doing this in lab too. Widow's peak is dominant and a not widow's peak is recessive. So all these individuals who have a widow's peak are expressing the dominant trait. Now, what do we know about their genotype? If they have the dominant trait, the dominant phenotype, what do we know about their genotype? It could be, let's use D's because W's are terrible letters. So having a widow's peak is dominant, so that's a capital D. No widow's peak is recessive. So if someone has the dominant phenotype, what do you know about their genotype? They have to have at least one dominant allele. So they could be homozygous dominant, or they could be heterozygous, we don't know. So I always start with the recessive ones. In this case, if I know it's a dominant trait, all these ones that don't have it have to be recessive, right? If they don't have the trait, they have to be recessive. So I just write their genotypes below them. And then you can work backwards. From what you do know. So let's start with this one. We know this one is little d. So where did this kid get their little d's? They had to get it from their parents. So this parent has to have a recessive allele and this parent has to have a recessive allele, but we know that they have to have a dominant allele too, because they are affected by the trait. So we know they have to be heterozygous because they produce an unaffected offspring. What do we know about this kid? It has at least one dominant allele. Could it have two dominant alleles? It could. And can we tell? There's no way to tell, so you just have to write both possibilities. So sometimes you can't tell for sure. Okay, this one we know is little d. Working backwards, we know that this one got one little d from dad. Where did it get the other little d? From mom, so mom has to be heterozygous. Same thing here, we've got these two kids that are both little d. We know that one little d came from mom, the other little d had to come from dad. So dad has to be heterozygous. What about this one? Has to be what? because the only allele it could have gotten from its mom, her mom, is a little d. So it has to be heterozygous. So in this case, we're able to figure out for sure what everybody is except for this one individual down here. Okay, so I gave you 2 pedigrees. You didn't get one, I'll bring you one. Let's start with the one that has the numbers on it. Okay, start with the one that has the numbers on it. See if you can figure out first, is it dominant or recessive? And then see what you can figure out. Oh, shoot, we only have 1 minute. We don't even get to finish. Okay, take this with you. We're gonna do these together at the beginning of class next time, so bring them back.